Nervous tissue is composed of two fundamental and distinct classes of cells. These are neurons, which are the excitable signaling units, and neuroglia, also commonly referred to as glial cells, which are the essential non-excitable support cells. Neurons possess the unique ability to generate, propagate, and transmit both electrical and chemical signals, forming the basis of all neural communication. In contrast, neuroglia, while not electrically excitable themselves, vastly outnumber neurons by a ratio of at least 10 to 1. They perform an array of critical functions that are absolutely necessary for neuronal survival and optimal function, including maintaining chemical homeostasis, providing physical structure and scaffolding, and producing insulating sheaths around neuronal processes. Understanding the detailed anatomy of a neuron is key. The cell body, or soma, serves as the metabolic and biosynthetic center of the neuron. It contains the nucleus and prominent clusters of rough endoplasmic reticulum called Nissl bodies, which are responsible for intense protein synthesis. The soma integrates the multitude of incoming signals. Extending from the soma are dendrites, which are highly branched, tree-like extensions specifically designed to receive incoming signals from other neurons. Their extensive branching dramatically increases the surface area available for forming synaptic connections, allowing a single neuron to receive input from thousands of others. The axon is a single, long, cylindrical extension that functions as the neuron's output cable. It is specialized for propagating electrical impulses, known as action potentials, away from the cell body toward other cells. The critical region where the axon joins the soma is called the axon hillock. This area has a high density of voltage-gated channels and serves as the trigger zone where action potentials are initiated if the summed incoming signals are strong enough. At its far end, the axon typically branches into numerous fine terminals called axon terminals, or terminal boutons. These swollen endings form the presynaptic part of connections called synapses and are packed with vesicles containing neurotransmitter chemicals. Neurons are classified structurally based on the number of cytoplasmic processes, or neurites, extending from their cell body. Multipolar neurons are the most prevalent type within the central nervous system. They possess a single axon and multiple, branching dendrites emerging from the soma. Examples include motor neurons in the spinal cord and the vast majority of interneurons in the brain. Bipolar neurons have a very distinct morphology, with one single dendrite and one single axon extending from opposite poles of an elongated cell body. This configuration is characteristic of neurons in special sensory pathways, such as retinal bipolar cells and olfactory receptor neurons. Unipolar neurons, more accurately termed pseudounipolar neurons, feature a single, short process that almost immediately divides into two longer branches, one extending toward the periphery to function as a sensory receptor and the other traveling into the central nervous system. The cell body of these neurons sits off to the side of this continuous process. This type is found exclusively in sensory ganglia, like the dorsal root ganglia next to the spinal cord. Functionally, neurons are classified by the direction in which they conduct nerve impulses relative to the central nervous system. Sensory neurons, also called afferent neurons, are specialized to detect stimuli from the internal or external environment. They transmit these sensory impulses from receptors in the skin, muscles, and organs toward the central nervous system for processing. Motor neurons, or efferent neurons, carry command signals away from the central nervous system to effector organs, namely muscles and glands, to elicit a response such as contraction or secretion. Interneurons, also known as association neurons, are confined entirely within the brain and spinal cord. They form complex networks that connect sensory and motor neurons, enabling integration, processing, modulation, and the execution of reflex arcs and higher cognitive functions. The neuroglia of the central nervous system consist of 4 primary types, each with a highly specialized role. Astrocytes are the most abundant and functionally diverse glial cells. Their star-shaped morphology with numerous radiating processes allows them to perform many duties. They form a structural scaffold that supports and anchors neurons. Astrocyte end-feet processes envelop capillaries and are crucial components of the blood-brain barrier, tightly regulating which substances can enter the neural tissue from the bloodstream. They are master regulators of the extracellular environment, actively mopping up excess potassium ions and neurotransmitters like glutamate after synaptic transmission to prevent neuronal overexcitation. Astrocytes also provide direct metabolic support to neurons, supplying them with energy substrates like lactate. Furthermore, in response to injury, astrocytes proliferate and form dense glial scars, a process that can both seal off damaged areas and unfortunately impede regeneration. Oligodendrocytes are the myelinating cells of the central nervous system. They have fewer, broader processes compared to astrocytes. The primary function of an oligodendrocyte is to extend its plasma membrane and wrap it concentrally around segments of multiple adjacent axons. This wrapping creates the myelin sheath, a thick, lipid-rich insulating layer that is essential for rapid signal conduction. A single oligodendrocyte can myelinate segments of dozens of different axons. Microglia are the resident immune cells and phagocytes of the central nervous system. In their resting state, they are small cells with highly branched, motile processes that constantly survey their microenvironment. Upon detection of damage, infection, or debris, they rapidly activate, retracting their processes to become amoeboid, mobile cells. They then phagocytose, or engulf, cellular debris, dead neurons, infectious agents, and even prune unnecessary synaptic connections during development. While protective, chronic microglial activation is a key contributor to the harmful neuroinflammation seen in many neurological diseases. Ependymal cells form a simple epithelial lining. They are cuboidal to columnar cells, many of which bear motile cilia on their apical surface. These cells line the fluid-filled ventricles of the brain and the central canal of the spinal cord. Their primary functions are to secrete cerebrospinal fluid and, through the coordinated beating of their cilia, to help circulate this fluid throughout the ventricular system, providing cushioning and a stable internal environment for the brain. In the peripheral nervous system, the supporting neuroglia are represented by two main cell types. Schwann cells are the principal glia of the periphery and are analogous to oligodendrocytes, but with a key difference. A myelinating Schwann cell dedicates itself entirely to a single segment of one axon, repeatedly wrapping its own membrane around it to form a compact myelin sheath. Non-myelinating Schwann cells also exist. They ensheath multiple small unmyelinated axons by enveloping them within grooves on their surface, providing vital metabolic and physical support. A critical feature of Schwann cells is their ability to create a regeneration-friendly pathway, guiding axonal regrowth after injury, a capacity largely absent in the central nervous system. Satellite cells are specialized glial cells found specifically in peripheral ganglia, such as dorsal root and autonomic ganglia. They form a flattened, enveloping layer around the neuronal cell bodies within these ganglia. Their roles are to provide structural support, electrically insulate individual cell bodies from one another, and help regulate the delicate chemical and ionic microenvironment immediately surrounding the neuron, controlling nutrient and waste exchange. The process of myelination is fundamental to nervous system function. Myelin itself is not a distinct substance, but rather a massively extended and compacted glial cell plasma membrane, extremely rich in lipids. Its formation by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system creates periodic interruptions in the insulation called nodes of Ranvier. At these nodes, the axonal membrane is exposed and densely packed with voltage-gated sodium channels. This anatomical arrangement enables saltatory conduction, where the action potential does not travel continuously along the axon, but instead leaps rapidly from one node of Ranvier to the next. This mechanism conserves energy for the neuron and increases conduction velocity up to 100-fold compared to unmyelinated fibers of the same diameter. Communication between neurons occurs at highly specialized junctions called synapses. A typical chemical synapse consists of three parts. The presynaptic terminal of the signaling neuron contains synaptic vesicles loaded with neurotransmitter and mitochondria for energy. The synaptic cleft is the narrow extracellular gap, about 20 to 40 nanometers wide, separating the cells. The postsynaptic membrane of the receiving cell contains embedded receptor proteins that specifically bind the released neurotransmitter. The sequence of synaptic transmission is precise. An arriving action potential depolarizes the presynaptic terminal, opening voltage-gated calcium channels. Calcium influx causes synaptic vesicles to fuse with the membrane and release their neurotransmitter into the cleft. The neurotransmitter diffuses across and binds to receptors on the postsynaptic membrane. This binding directly or indirectly opens ion channels, generating a local change in membrane potential called a postsynaptic potential, which may be excitatory or inhibitory. This cellular knowledge has direct clinical importance. An astrocytoma is a primary brain tumor that arises from the uncontrolled growth of astrocytes. Multiple sclerosis is a classic autoimmune demyelinating disease where the body's immune system mistakenly attacks the central nervous system myelin produced by oligodendrocytes, leading to disrupted nerve conduction and a wide array of neurological symptoms. Guillain-Barré syndrome represents a similar process in the periphery where an immune response targets the myelin sheaths formed by Schwann cells, often causing rapidly progressive muscle weakness. In major neurodegenerative disorders like Alzheimer's disease and Parkinson's disease, reactive changes in both microglia and astrocytes, a state often termed neuroinflammation, play a significant role in disease progression by releasing harmful cytokines and contributing to neuronal damage and loss. In conclusion, the nervous system functions as an exquisitely integrated unit. Neurons are the excitable, information-processing elements with remarkable structural and functional diversity. Neuroglia are the indispensable non-neuronal partners that provide the essential infrastructure, structural integrity, electrical insulation, immune surveillance, metabolic support, and environmental homeostasis. The seamless operation of the entire nervous system emerges from the intimate and interdependent relationship between these two foundational cell types. Thank you for watching. Don't forget to like, share, and subscribe. See you in the next one.